cell development, even though it's not as complex as T cell development, you need to know what are the stages of B cell development, where do they happen? They obviously happen in the bone marrow. And what are the, and what is the role of, for example, what is the role of the endogenous cells in the bone marrow? And so basically, first you need to know is these cells develop in the bone marrow and then they mature. They continue to develop and mature in the spleen. This is not to say that without a spleen you can't get mature B cells, but when we talk, this is the normal developmental process. So I just want to point out that if you have a splenectomy, you still have B cells. You can still get mature B cells. They can actually mature in the bone marrow. It's not the normal chain of events that happens. The normal chain of events is you get B cells that develop in the bone marrow that become these immature B cells. They go out into the periphery, circulate to or through the spleen, and then they will spend some time in the spleen becoming mature cells. And if you really think about it, T cells, their negative and positive selection happens in the thymus, but the thymus is so specialized because it's producing all of these antigens. It's the perfect environment to kind of do that negative selection, whereas T cells, the bone marrow doesn't express everything that is found in the body. And so you want these immature cells to go out and still kind of be in a state where if they see antigen, they are going to get this negative signal out.
What's the general cycle of the become everything focus on it?
Nope. That's where we're starting. So, what's important to know is where the B cells develop, that development continues outside of the bone marrow, where they will eventually mature, and like I said, the majority of the time they're going to mature in the spleen or in the other secondary lymphoid organs like the lymph nodes. What we're focusing on is the spleen, though.
So what are other secondary?
Lymph nodes. So spleen, lymph nodes. So the only thing I need you to know from this is there is a stage, there is this alternative, there is B cell development still before we even have bone marrow. The first place that occurs is in the yolk sac, and it's an alternative type of B cell development because the environment is a little different, and we primarily get these type 1, so we get these B1 cells. It's important to know where that starts. Some of the questions were at what stage, at what day in development, where would you find B cell development at what day, but I'm not going to get into that detail because I don't think it's going to be that important.
So what do you need to know? You need to know.
You need to know that there's B cell development in the, like I said, there's B cell development that happens in the yolk. That's the first place it happens, in the yolk sac, that you get B cell development even before you have bone marrow, and that it's an alternative, doesn't form B2, but B1 type of cells, and those B1 cells are analogous to gamma delta T cells and T cells, so they're going to be more, they're not going to have the same diversity, and they're not going to, and they're also going to be specific for carbohydrates that are on, that are known carbohydrates on pathogens. So they're kind of a little bit, they're more part of the innate immune system.
But when you said the fetal liver, I thought you said it's
You get cells that start from there,
yeah.
and then, well, the AGM goes and becomes the kidney. I didn't say that so the AGM eventually will become the kidney, but then you start to get, but you don't only have cells produced in the AGM, then you start to, the fetal liver starts to be involved, and the bone, then eventually when they get bone marrow is involved. So for example, on day 11, you would have B cells being developed in the placenta, in the fetal liver, hematopoiesis occurring. They're not only making B cells, but they're making red blood cells. They're making different, again, because the environment's a little different, they're actually making red blood cells that are nucleated red blood cells. And as you might remember, most of our mice and humans have anucleated blood cells in a mature mouse or a mature human, whereas if I take a cord blood and I take a cord blood and I look at the red blood cells, the majority of them are actually going to be nucleated, and that becomes an issue if you're trying to get a buffy coat from a cord blood. It's really hard because instead of having these nice, light, not dense, actually they're not as dense. So the red blood cells, they act a little differently. So when you take blood from the cord blood and you try and spin it down, the red blood cells kind of don't separate from the white blood cells very well. I just
So they don't form the buffy.
Yeah, it doesn't form as nice a buffy coat. So you usually have a lot of, and also when you go to lyse the red blood cells, lysing. So the lysis process, if you've ever done this in the lab, red blood cells that don't have a nucleus lyse really nicely in the lysis solution, whereas nucleated red blood cells don't lyse very well. So
Because they have membrane.
because they're a little bit more intact, and they're when you get rid of the nucleus, something about that process makes the cells more susceptible to the lysis.
That's called a hematopoiesis because mostly, because you have to go to the retinal side before, that's why they move out of the bone marrow, right? The red blood cell.
Yeah.
So they produce in bone marrow, right?
Red blood cells are also produced in the bone. And we'll cover that a lot in the talk.
What's a book? You still didn't.
No, I don't know. I'll look it
The
up.
book costs $80. You can give us the online.
You don't need a new book.
Even the old book is $80. I don't have money.
So what you need to know again is that early development, before you have bone marrow, the B cells that are produced are these B1 cells. And what are the B1 cells? Like the gamma deltas, they don't have a broad repertoire. They're going to respond to carbohydrate antigens found in pathogens. So these are the type of things that you might have a question on. What's the stromal cells? So these stromal cells are these matrix cells, these supportive cells. What is the role of stromal cells in the bone marrow? And the two important things that they do are they express things like CXCL12, and CXCL12 is going to basically maintain those precursor cells in that area where they can become mature. So it's going to, these act to localize the cells within the area of the bone marrow where they can get signals like IL-7. So the two roles of the stromal cells are going to be to localize B cell precursors in areas where they can become mature B cells, and they can get the proper signals in one of those signals. So the second role of the stromal cell is to provide those maturation signals like IL-7. So they will localize and give signals, and so IL-7 would be the major signal that's going to drive B cell differentiation, whereas with T cells, the signal is Notch. So Notch is expressed in the thymus, and that's driving T cells. So if we, for example, knock out Notch in cells, the Notch receptor, then we're going to get B cells. If we overexpress the Notch receptor, a constitutive form of the Notch receptor, then we're going to get T cells. And so that Notch is kind of the deciding factor. But IL-7 is kind of the deciding factor for B cells to mature.
But if you knock out, if you don't have the IL-7, do the B cell become T cell?
They still need that Notch signal to become a T cell. So you would have an image, you would have a cell that wouldn't go through VDJ recombination at all. They need some signal to kind of tell them, hey.
So only Notch can do that.
Notch or IL-7.
The
So
IL-7
they're looking for some signal to tell them to.
Even for B B cell,
Yeah.
right?
So here I put, this is the slide that is new for this review. I didn't want to add things, but I thought this was a nice breakdown of what's happening where. Okay, so you're in the bone marrow during these stages, during the stem cell, the pro-B cell, the pre-B cell, and the immature B cell then goes out of the bone marrow into the periphery, goes to the spleen or the lymph nodes, and then you get further maturation. And so the things you need to know in this are how do they respond to antigen where, and also what IG do they express at what stage in development. So you have IgM and IgD. So those early immature B cells are going to produce, they're going to have membrane-bound IgM is going to be the only Ig they present on their cell surface. As they become mature, they start to have both IgM and IgD expressed. And then as they become plasma cells, those plasma cells will then make things like IgG and other IGs. But right now, through this, the important IGs to know for development are IgM and IgD, and when do you see those? Because those are key factors in determining what type of B cell you're looking at, mature or immature. Also, those are important in identifying energic B cells, which is the third type of B cell. So we have B1s, B2s, and B3s, and B3s are more energic because these are B cells that were successful in making a B cell receptor, got out into the periphery, but are have seen self-antigen, are specific for self-antigen out in the periphery.
What's the difference between B1 and B2? So B1 is
B1 is as I discussed, yep.
So B2 is
And
mature.
B1s also will continue to proliferate out in where B1s localize also is a little bit different. They localize in the peritoneal and the cavities. So in the also in the
Oh, so you mean
pleural
near the gut,
cavities,
near the gut.
in the gut, and around the lungs. So the pleural cavities, peritoneal cavities, all of those cavities are where you will find the majority of those B1 cells. So where they localize also is a little bit different. So the majority of B cells you find in the peritoneal cavities, the pleural cavities, are actually B1s as opposed to B2s. And that kind of makes sense. So B1s are what? Innate. They're kind of these innate cells that are looking for any type of pathogens that come in. And a lot of times the innate response is our first response to pathogens. It's a very quick response. We know that these innate cells, we know they're not specific to self because we don't make that type of sugar. And so those will respond very quickly. So it makes sense that the B1 cells, these innate cells, will basically line any area where they're going to line the gut. So we have lots
So
of
how about
pathogens.
here? So
They line the lungs. So we're breathing. In the pleural cavities are going to surround the lungs, and so they're going to be early responders to any pathogens that get through that path. So you would find the majority of the B1s there. B2s you would find in the blood. You would find in the spleen, in the lymph nodes. So places like that. And then B3s, you don't find a whole lot of them, but that is another type of B cell. These are B cells that were not negatively selected in the bone marrow, that got out, that were specific to self, but then they saw lots of self-antigen in this state where they were still in this immature state. So they were in this immature state, they got out, but then they saw lots and lots of antigen before they became mature, and they became energic. They didn't delete, they didn't...
what's energetic?
Energic means that they won't, when they see antigen, they don't start proliferating. They don't respond
What's
like
the word you
normal.
have?
Energic?
Atmospheric.
I think I have that later on. So here's, if you like pictures better, this is just a picture of the bone marrow, what stage of maturation you are at. So again, good to review that.
Excuse me.
Yep.
Before you go further, when this way, a lot of autoreactive antigen.
Yep, and I'll talk more about that in a slide, about 4 more slides now.
That sounds like good.
So B cell development, I've got a video. Again, these slides are up. They're called Chapter 10 Modified. And this is, we don't have time to watch the video, but it is a nice video where he goes through the different stages of B cell maturation. So if you study that on your own, watch the video, it might help with more visual way to remember B cell development, because that's what you're going to be responsible for, is knowing the different stages of B cell development, what they're called, and what IG you're going to see on them at what time, where they are, and that B cell receptor development. So did BJ recombination occur in what stage? Did BDJ recombination occur by this time? Do you see the mature IG at this stage, or do you see mature light chain, or do you see what's the other light chain? The fake light chain.
Oh, great.
Yeah.
Great.
Uh-huh. So, what B cell receptor do you see? Do you see the surrogate receptor, the light chain, or do you see the mature light chain? Yes.
Where do you get that in the new B cell receptors?
So here it's going to tell you.
Oh, that. Okay.
It's going to say what recombination. If light chain hasn't been recombined, here you have your picture. So this pre-B cell does not have the mature B cell receptor. It has the surrogate light chain still. So that's you need to know, just based on these pictures, when do you first get the mature B cell receptor? And
When do you get B cell receptor? In a pre-B?
So pre-B, you still have the surrogate light chain, and then immature, the actual immature B cell stage is when you would have the complete light chain.
So immature or mature? Mature.
Immature.
Immature, you have a copy.
Yeah.
So
And you want that, if you think, just think about it logistically, the immature cell is still going to go through negative selection until it becomes a mature cell. So any signal that immature cell sees, the signal that it gets. And the reasoning, we think we understand how you can get one signal in an immature and a different signal. So they're both going to see antigen, right? So if the immature cell sees antigen, it's going to cause a signal to induce apoptosis or antigen. When you get a mature cell, it's going to see that receive the same antigen signal, but it's going to proliferate. And the reasoning they believe is because when you look at the immature cell versus the mature cell, the amount of cholesterol in the cell membrane is markedly increased in the mature cell, where it's much less in the immature cell.
why?
And cholesterol plays a huge role in lipid rafts. And lipid rafts are where, if you remember, and we'll talk about lipid rafts again when we talk about T cell activation, but when we talk, lipid rafts play an important role in localization of lots of receptors. So if you don't have lipid rafts, you get very diffuse receptor location on the cell membrane. They don't come together and localize in an area. So if you don't have cholesterol, you don't have lipid rafts. If you don't have lipid rafts, you're going to have very diffuse receptors kind of dispersed all throughout the cell membrane. And so the type of signal that you're going to get is going to be a different type of signal because of the localization of the receptor itself. So if I localize all the receptors, and that plays a huge role also in the strength of signal that you get, because a lot of times when you localize receptors together, those receptors will have enzymatic activity with each other, like we had in the T cell receptors, which have ITAMs that will become phosphorylated. So if they don't localize next to another receptor, you don't get as much phosphorylation of the receptor itself. And so the type of signal, in a sense, can be different. It can go through a different pathway because of that.
Before you go, immature, the naive, and mature. What's the difference between immature and naive?
Naive is typically a
You
word
don't see anything.
we talk about with T cells.
Oh,
And talking about T cells, use the term naive, whereas when
T
we're
cell is not you.
yeah, when we're talking about B cells, we usually talk because it's a specific stage. It's an immature stage. So the words naive is a similar term, means a similar thing. It's a but you shouldn't use them interchangeably because a naive T cell is a mature T cell. It's still a mature T cell, developmentally speaking. Okay, so B cells, we consider them immature because they're still developing, they're still undergoing positive and negative selection at this point. Whereas the T cell, a naive T cell, has gone through positive and negative selection, it's exited the thymus, and it is no longer going through negative and positive selection. Now, granted, it's a naive T cell, if it doesn't get the proper signal, will die, but it's still what we consider a mature cell.
So
Developmentally
you have a recent immigrant some cells, CAR T cells, when they have a single quality. So what's the difference between a naive and that immigrant, recent immigrant T cell?
Naive is going to be, those are different terms you're talking about.
They just get out of the cells.
Yeah, so that's the stage that they're at. They just got out of the thymus, and they may be have a small window where they don't respond as strongly to antigen. They're still a mature cell, but they are kind of attenuated a little bit, and so yeah. But naive, they're still naive until they receive the proper signal. So those are kind of two different things. A lot of times, even at certain stages, you can induce a cell that is activated if you get the proper signal. So naive just means you haven't seen antigen yet. But then again, that's still a term we talk about. When we talk about T cells, we talk about naive. When we're talking about B cells right now, we're not. We're using the term immature. And we're still developing. So we're still undergoing, we can still undergo negative selection. And another thing, in that same vein, if it's immature, it may be possible still for it to choose a different light chain, right? So as long as while you can still manipulate the chromosomes or the chromosomal DNA, you haven't matured. You haven't become a mature cell. Whereas the T cells, a naive T cell, it's not going to change its receptor.
So for a B cell, if you mature, they can still choose the alpha?
So it can still choose a different light chain, for example. Yes, in that immature state. And they've done, I don't know if I still have the slide on here, but there are studies where they showed that if you have, you can still, yeah, I actually took it out. But you can have a heavy chain that is specific for a self-antigen. But once it gets out into the periphery and sees that antigen, it will actually undergo, it will choose a different light chain and that doesn't recognize self-antigen. So you'll still see that heavy chain that actually made that same heavy chain can be used to make a non-autoreactive B cell because it's going to choose a different light chain.
Oh, I see.
But that's going to happen, that can happen during its immature state. So there still is modifications that can occur within the cell until it becomes a mature.
So that's when you say that you mean is they have 4 chance choose the immunology?
Yep.
Right? Is that what you mean for even after
Yeah.
immature?
Yeah.
Because
But I'm not going to cover really that on the exam, this exam. That was chapter
4, 5, 7.
yeah. So I'm not going to go rehash that. But so earliest steps in. So now again, the cells, you need to know what are the precursor. You got the hematopoietic stem cells. That's easy enough. Then you get these multi-potential progenitor cells. These,
Where is that?
so right here. Then you get these LMPPs. lymphoid primed multi-potential progenitors. These are all before they've decided to become a B cell. Then you get these early lymphoid progenitors, and then this common lymphoid progenitor. And so here's HSCs. You don't need to memorize what are the different markers, although it is good to know that they express c-KIT and SCA1. The important thing is this first bullet, self-renewing and multi-potential. But, all of you probably already know that.
So what's the first one?
What makes a stem cell is that it's self-renewing and has multi-potential. So the MPPs are the next stage in development. They lose the capacity of self-renewal, so these are no longer stem cells because they can't self-renew, but they still can become lots of different cells. They begin to make CXCR4 that helps them remain, that's an important one, in the bone marrow. Then you get these LMPPs, and these express FLT3 and start to produce IL-7 receptors. So they have now the receptor, yeah.
Is there a key ratio between the HSCs and the MPPs, or is it more that
What's
as the
MPP?
number of MPPs
What's
mature into the LMPPs?
All of that is regulated. Part of all of that's pretty tightly regulated. The ratio, the ratios are pretty well regulated. I couldn't tell you what the ratios are. It is, there aren't a whole lot of stem cells compared to other cells, but
There's
it's hard to say.
greater cells.
But it's not 100% a ratio thing either.
So, before you go to that part,
Like the developmental process,
Yeah, there are other factors that figure into ratios. That's why we all actually have different ratios of things depending on genetics sometimes and depending on certain other events, and age as well. So those ratios don't stay the same over age as well. So there are other factors that play into those ratios. But things that you need to know for LMPPs are they start to make IL-7 receptor, and that now you need that to become a B cell. ELPs, now they start to express RAG1 and RAG2 that we need for recombination events to occur in the chromosomal DNA, and then the CLPs, now they've lost their myeloid potential but retain other potential. So this is a good slide. You need to kind of know what are the different stages before we even get to that pre-pro B cell, and at what stage and what potential do the cells have at different stages. For example, a common question is at what developmental stage can cells still become a T cell or a B cell? And so the CLP would be that last stage where you can either become a T cell or a B cell depending on the signal you get. So up until the CLP, these cells are still multipotent in that they can become T cells, B cells, NK cells, dendritic cells. But they are, again, not self-renewing. And so this, again, it would be good to kind of have this down, what cells
so
can happen, or what the
do we
stage.
need to remember all these stages?
But only in the basic form of what is the order that you would find them in, and what is the potential at any given level. And then once we become committed to becoming a B cell, okay, so now we start to enter that B cell development. It's important to know, again, this is going to be just reviewing what I had up on the other slide, where the pre-pro B cell, you do not have any IG expression on the surface yet because you haven't recombined anything. It's that early pro B cell where you start to get D and J recombination. The late pro, you get some BPJ recombination, and so you still don't see surface. So you see that pre-B cell receptor at the pre-B B cell stage, and then you only get mature IgM at that immature stage. And it's not until those cells mature that you start to see IgG. And so these are the difference. So you need to know that you go from pre-pro. I'm not going to break up early versus late pre-pro. We're just going to say there's pre-pro, then there's pre-B cell, and then there's immature B cell.
So you just need a pre-pro or later?
Yeah, I mean, I'm not breaking down, but if you say that the pro B cell, you start to get DJ or you get VDJ recombination happening in the pro B cell, then that'll be fine. So if I come back to this again, we've got a pro B cell, pre-B cell. So in the pro B cell, okay, you have
Nothing.
yeah.
Group B and nothing. I think that means it's just DJ.
So kind of you can go by this list to see where the recombination occurs and what IG is expressed. Again, I still have this slide on here, but I don't expect you to tell me what signaling occurs through the IL-7 receptor, but you do need to know that at what stage do you receive that IL-7 signal, and IL-7 receives a survival signal. It happens before VDJ recombination, so this is kind of driving now that VDJ recombinations had occurred. Strangely enough, you might see that it also induces notch, and so that might be a quandary for most people. And that kind of highlights also that when we say notch is needed for a T cell signal, that's not the only thing notch does. It's just if you have this notch signal early on, it drives T cells versus B cells. B cells start to make the receptor for notch. There are cells that express notch as they egress from the bone marrow, and they believe that that plays a role in, if you look at proliferation, I think that here we have whether do we have a picture? Does it say whether they proliferate? So proliferation, if you look at proliferation, we don't have a lot of proliferation happening from here on out, okay? And they believe that notch signal actually will kind of maintain those cells in a state of where they're not proliferating. So it's just, you don't need to know that, but it's just, if you had a question as to why do B cells make that notch receptor, if notch is involved in T cell, driving T cell maturation, the reason is because notch does other things as well, and it's involved in kind of suppressing proliferation of that immature B cell until it becomes a mature B cell. So we've got
Proliferation of pre-B or the immature?
say that again.
You said notch involve suppression of proliferation of
Yeah.
B cell in
Yep.
general?
Yeah.
Oh, just pre-B.
It's actually, yeah, in general, that's what we believe the function is in immature B cells. So pro B cells, you get DJ recombination is completed, and you start to promote B and DJ recombination. You get CD19, so CD19 is important to know that that's a B cell marker. So if you're looking at mature cells in the blood, CD3 is a marker for T cells, CD4 is specific for helper T cells, CD8 specific for cytotoxic T cells, CD19 would be your major, your B cell marker. So CD19 is not going to be expressed on T cells or other subsets of cells.
Can you see again, CD3 is marker for T cell?
Yeah, but we'll get into that later. I was just kind of highlighting that CD19 is really Now, the other thing to know, the thing you need to know is immature B cells in the bone marrow are sensitive to tolerance induction. So still, if they see signal, it's going to induce tolerance. It's not going to induce production of antibody or maturation. So those immature B cells, they're going to go to the secondary lymphoid organs like the spleen. The majority of them are going to go through the spleen, and then they're going to go through 2 stages. They're going to go through the T1, which is still immature, to the T2 stage, and this is just showing you that it's in the T2 stage of
Why do we call it T1?
these are not T cells.
Then why do you
Right
call
here.
it T1?
Because they want to confuse you.
That's good.
So these, so the major point here is the ratios of IgM to IgD. Again, the T1s have lots of IgM, low to no IgD, whereas the T2 stages you now see IgD expression. Where intermediate IgD expression, whereas mature B cells or B2 cells, you're going to see high expression of IgD and low expression of IgM. And if we had B3 cells, you would see no IgM, but we don't have
Where
that.
do you see that?
I see it off, you don't see it off the slide?
Mature.
It's not there, but. So the immature B cells come to the spleen. They will then enter the spleen through the splenic arterial vein, splenic artery, and then they will then localize to the follicle in the spleen. And so the T1 subset still undergoing screening. So here we're still undergoing screening for possible negative selection, and then when they transition to the T2 stage, they now start to receive a positive selection. So if they see, so this is where in the follicle, and so this is the first place in the splenic follicles is where these cells are now going to sit there. Now they've become mature enough to where they're going to sit there, and actually this is where if they receive a signal, now that's going to be a positive signal. This is where you're going to first encounter pathogenic blood pathogens. So again, our blood passes through, it's going to pass through the spleen, and our spleen is where we recycle and get rid of dying red blood cells. So these dying red blood cells, if they happen to have been infected with some pathogen, then that pathogen will be present in these areas, and so you have these, that's why we populate these areas with newly matured B cells that if they receive the proper signal now, if they receive a signal that will be a positive signal that will lead to proliferation. Yeah.
Is that image from the bone marrow?
So this is now within the spleen. So here's a picture of the spleen. Here's your splenic artery, and you have the perioarteriolar lymphoid sheath, which cells will come out into this peripals, and then they will then, and in the pals, they are still in that T1 phase as they progress to the germinal centers. These germinal centers will now be populated with more cells that become mature and activated cells. So just kind of going all the way back to, I think, chapter 2, where we talked about lymphoid organs and kind of the structure of those lymphoid organs. And so you have your white pulp and your red pulp, and the red pulp is where the red blood cells are being lysed, and macrophages are engulfing them and recycling that material. And also this is where you're going to have antigens present that are possibly pathogenic antigens, and so you then would want B cells populating close
center in a white cell?
Yeah.
So the B cell was there?
As they become activated, they move to the germinal centers, which is where you get, the germinal centers are gonna just like you have in lymph nodes, that's where B cells start to proliferate and start to produce lots of antibodies, become plasma cells, and those germinal centers, then those plasma cells will go out into the bloodstream, go back to the bone marrow, populate the bone marrow, as well. But, again, we're gonna cover that a little bit more when we talk about B cell activation. You just need to know where the B cells are maturing and becoming now. So the third type of B cell is the B3 cell, or the T3 B cell, and that's an energic B cell that is specific to a self-antigen, but because it's seen lots of self-antigen before it ever got to the mature phase, it doesn't make a lot of IgM, and apparently signaling through IgM is going to produce proliferation, which is probably why mature cells still make low levels of IgM. So if they see signal through IgM, that's going to signal for proliferation.
We don't have a list, right? We don't have that.
You don't have it?
We don't have a list.
Yeah, it's in there. It's a couple slides down because I took some slides out. So this is the experiment where they showed they took an animal that produced a transgenic. Anytime you have a B cell transgenic animal, that means that they put in a gene that makes antibody, so it's already undergone recombination, and so these mice make lots and lots of B cells that are specific for NA glycoside, and so you're going to find that in eggs, and then these cells, these mice make lots of NA glycoside. So these guys make antibodies for NA glycoside, and these make NA glycoside. We don't usually make NA glycoside. So then you get the double transgenic mouse now, is where they looked at that double transgenic mouse, and they still found B cells. They've got out in the periphery that, and this is what this is saying, is these have B cell receptors, because they took, let's just say this is GFP. So they took NA glycoside that was bound to a color, to green, and they showed that, oh look, we have cells that have receptor for that, because that NA, that hell-bound, they're hell-bound to the cell. But then they don't have IgM. So we have another antibody that binds to IgM. So these cells, if it's an animal that doesn't have, that doesn't make hell, doesn't make the protein, it doesn't make the antigen, they get lots of these mature, or we don't know if they're mature, but they do have IgM, and they also are specific for hell. Whereas if this animal makes lots of hell, you still get B cells out in the periphery, but they don't make IgM to any extent. So these are probably IgDs.
And we know they're not IgM.
Because there's an antibody for IgM that is on this axis, that is red, for example. So these are red and green, whereas these are only green. So the fact that there's no antibody that binds to IgM is saying that there's no IgM, because this would be over here if there was IgM. So these are specific to to hell, but they don't have any IgM, and so they've classified these cells as these energic cells. So they're not as functionally important because they're not supposed to function. So when these
What's energetic?
Energic means that right here, when we look at their, they're not producing antibody. So when they see antigen, so if we give them antigen, if we give these guys, so let's say we take these cells here and we give them antigen, you're going to get lots of antibody being made. So IgG, they're going to start making IgG. And remember, IgG isn't membrane bound, it's soluble, so they'll start making IgG, they'll become plasma cells, they'll start to proliferate, they'll make IgG, they're your B2 cells. But these T3 or these energic cells, when they see antigen, they can bind to antigen, because we just saw they can bind to antigen. They're binding to antigen. But when they bind to antigen, you don't get antibody, you don't get any IgG made, you don't get serum, you don't find antibody in the serum that's specific for health, and you don't. So basically, and they don't divide, they don't proliferate in response to health. So these are kind of, and that's what we consider allergic. They haven't died, they're not apoptotic. And the reality is they don't live as long. These cells don't tend to live as long as mature M2s. Yeah.
So do they have any function? Or they just kind of...
Do they have any function? If you take away the health, so if you take these cells, these immature, so if you take these immature cells and you put them in a mouse that doesn't have any hell in it again, so now we're taking these cells out and you put it in another mouse. As they mature, they become active B2 cells. So they don't necessarily have function, but they kind of are.
But they can still differentiate.
They can still differentiate. The earlier ones can still differentiate to become activated. So we may have developed these to where, let's say you're making, they're still kind of there. They're still, hey, if this really wasn't self-antigen, so if I stop seeing this antigen, maybe it's something I ate, Maybe I ate something with a lot of that antigen. And so once I stop eating that, maybe they're going to start responding to it. And that's maybe how allergies may develop. We see a lot of antigen, we get these guys, but then we take the antigen away and we bring it back, and then now you have these guys that are active in an active form, and they're going to start. And maybe that's why I'm totally conjecture that could be like a peanut allergy thing, for example, where if you they're actually starting to say that you should give children peanuts, because if you give it to them early on, before they
become tolerant.
Yeah, they become tolerant. So they're actually, they used to tell you don't give peanut butter to infants, and now they're actually saying give peanut butter to them like day one, give them peanut butter. So yeah, so our understanding of immunology is changing. Right. So let's see, what else do we need to know? Mature cells migrate to the lymphoid follicles. They express IgM and IgG. Again, there's so the follicular. These are our B2 cells. These are the ones we think about when we think about B cells. But remember, we have these B1 cells. So this is a comparison of our B2 cells, our B1 cells, and then we have another population of kind of these marginal zone B cells. So
What's that? That's not the.
so they populate, these guys populate the marginal zones of the spleen. If you go back and look at your spleen, the marginal zone is going to be, marginal zone right here. So it's kind of in the periphery. So these guys kind of populate this outer portion, this marginal zone of the spleen. And before we even go back and look at what do they do, you might say again, the T1 cells or the B1 cells are populating the peritoneal cavity, the pleural cavity. That's, they're going to be direct contact with things that come into your body, pathogens. Whereas here, this is in the marginal zones of the spleen. That's really close to where, very close to where the blood is going to be going through, the blood cells are being lysed, and so it might also have some innate function, because again, we populate areas that come in direct contact with the outside world or with potential pathogens. We populate them first with innate immune cells, the first defense. And so if we go back and look, where was that stuff? These guys aren't as, they are variable. It's somewhat, let's see, they respond to carbohydrate antigens similar to the B1s. So these again, are a little more varied than the B1s, but they may tend to respond to, they're kind of more, a little bit more innate, I guess. They're kind of but again, the important one to know is this molecular, but here it's important to kind of know the difference between these guys. So this would be a good one to say, to just look at and just know what is the difference between these B2s and B1s. Where do they look? Where would you find each of these? So that might be a good question. Probably focusing on B2s and B1s. But again, where would you find B1s? Where would you find B2s? And again, this is just reviewing what are B1s, why do we have B1s, why do they have less diversity? Because for one thing, you're not generating TDT, so you're not expressing TDT, so you're not adding, you have no end region addition. You're also so you don't have as much diversity, and these are more specific to particular antigens. 30 to 50% of the B cells in the pleura and the peritoneal cavities are these cells. So it's important to know where these cells are, and that the majority or a large percentage of the B cells in those cavities are these cells. So the pleura is the cavity surrounding. So if I take a balloon and I shove my hand into the balloon, all the space inside the balloon. So my fist is the lung. I push it into the balloon. You have this membrane surrounding your lung. So inside that is filled with fluid, and all that fluid has B cells. It forms to create a lubrication because your lungs are always expanding and contracting. So you don't want cells rubbing because you'll get inflammation, you get damage. You don't want that. And so you have a lot of B cells that will populate that. Also, in case you have a pathogen that comes in through your lung, you have early responders that can respond to that. So it's full of protein and antibodies and things like that. So the pleural cavity, just like the pleural cavity, we have peritoneal cavities in our surrounding all the organs of our gut. And the peritoneal cavity is actually because you can have lots of antibody-producing cells, and that's where I don't know if they still do it, but they used to make antibodies in mice, and they would inject B cells, clones into mice, the peritoneal cavity, let them grow, and then collect them from the peritoneal cavity of mice. Just as a side note.
The bloodborne recognition of HE, is it silver?
Say that again?
I think silver, isn't it? Bloodborne recognition. So when I first got that, it said silver. And then cell
fourth
pages.
bullet point.
Yeah, the fourth one, just bloodborne HE recognition.
Antigen.
Antigen.
Yeah, bloodborne antigen recognition. So any pathogenic antigen or antigen in general that's in the blood, that's so then again, comparing B cell and T cell that's important to know. They both have rearrangement of gene segments. They both have screening to avoid self-reactivity. They have small subsets with discrete function, like gamma deltas versus our B1s. They have larger general purpose subsets like the alpha betas and like our B2s. The location of maturation is different. Positive and negative selection occurs in T cells, whereas only negative selection occurs in B cells. And then T cells require presentation in B cells do require T cell help to secrete antibodies, but they don't have MHC restriction like T cells do. I guess here's more of again, just what we just went over. what are the differences between B and T cells. So you can go through that on your own, and that's pretty much everything. So again, what I would focus on, I wouldn't spend a whole lot of time other than what are, you know, it is, but it's not a, you know, where's the first place you start making B cells in the yolk sac, and these are B1 cells. B1 cells are different than B2, they don't have a lot of variety. B cells develop in the bone marrow, not in the thymus. The important cells that help to maintain those precursor cells are the stromal cells. They have two roles to localize B cell precursors in the area of the bone marrow where they're going to mature, and also to provide that differentiation signal, that IL-7 signal. And then when do you have what IGs are on what stage of development, and what recombination has occurred during what stage of development? What are the ratios of IG and IgD, IgM and IgD? What are the precursor cells from hematopoietic stem cell to pre-pro B cell? And then what are the stages? Again, this is just statement similar to this guy here, and then go through, and you don't need to know what signaling happens through IL-7, just that is akin to notch signaling to drive T cell versus B cell. So IL-7 would be the key signal you need to make a B cell. Immature B cells go to the spleen where they become mature. We've got T1 and T2. T1 is still going to receive negative signals or have negative selection, whereas T2 now, you're going to start making some IgD and IG, so low levels of IgD, and then once you become a mature cell, you're going to make higher levels of IgD and still some IgM. And then just, again, just a review of what's the difference between B1s, B2s, and the marginal zone B cells, which aren't as important, and then comparing B and T cell development. And so it seems like a lot, but I would think that just reviewing these slides would probably be sufficient to prepare for this portion. I'm only going to have probably 5 or 6 questions for this chapter.
So how many questions do you have total?
We'll have 50 to 60 questions.
So 30 questions from chapter 9, something like that?
No, chapter 1 will probably have like 15 or so questions, and then we'll have an equal number probably for chapter 9 will have like 15 or so questions, so 9, 10, then 11 and 12 will have a lot more. So I'll try to get to 11 and 12. We might have to push back. Do you guys know when your next exam for
It was supposed to be next Wednesday, but she pushed it back because she saw your test is next Wednesday.
Okay, well, we're both going to be jogging for exams. Okay. So if I don't get to chapter 13, I won't.
Not a
do
30.
that. 12. No, I'm going to get to chapter 12. We're going to cover chapter 12 next. Chapter 11. Sorry. If I don't get to chapter 12, we won't have it. So I've got 10 more minutes. So chapter 11 is going to be T cell activation. Now we get into the fun stuff, and this is really kind of the more meat and potatoes of immunology. We've spent all of this time telling you how you get B cells, how you get T cells, how they mature, what signals are involved in making a mature cell. Now it's time to talk about, because most of us take that for granted, obviously. People who have genetic defects don't take it for granted, but for normal people who are healthy and don't have any genetic defects in their process, we care a lot about remember, we're switching gears to T cells. They have to see antigen presented via MHC, and this is what's considered a first signal. So the first signal they get will be that antigen presented via MHC, and it's really important to know there are other molecular interactions that can provide a second, which is required activation signal. So T cells always require, they say always require 2 signals. In truth, they require kind of 3 signals because the third signal tells them what to become. The first 2 signals tell them that it's okay to become activated. If they only get 1 signal, they tend. Well, the first signal is going to be antigen presentation, and we'll talk about the second signal is going to be co-stimulation. So the first signal is going to be antigen presentation. Second signal is going to be do they receive co-stimulation, and then the third signal is going to be cytokines, the milieu of cytokines that they receive or see to help differentiate them to become different types of helper T cells. There's lots of different kind of helper T cells and possibly potentially different types of cytotoxic T cells as well. So once activated, T cells differentiate into their effector form, what we call effectors. So the effector form are going to go out and act. They're going to respond to stimulus and either kill cells in the case of cytotoxic T cells, or they're going to become helper cells and start making lots of cytokine and help to drive cytotoxic T cells if they're CD4 or helper cells. And we focus primarily on activation events overall, followed by differentiation. So first we're going to talk about activation, but then we're going to talk about once the cells become active, how do they differentiate to become different types of cells? Because there are a lot of different subsets of helper cells, or they can become suppressive cells as well. Okay, so again, T cells, CD4s and CD8s were shown seeing antigen being presented by a professional antigen presenting cell because that's the optimal way to activate cells. For one, the antigen presenting cell is going to present an exogenous antigens that's taken up. If CD4 comes along and sees exogenous antigen, it's going to induce that exogenous antigen to actually enter through the cross-presentation pathway and be presented via MHC class 1. So you can get an APC that is both activating CD4s and CD8s. And the nice thing about an antigen presenting cell is it has, if you see here, if you really focus in, we have our T cell receptors, but then we have these other guys over here. So we have T cell receptor and MHC and antigen.
There's no.
Oh, no pen. Okay, let me get the laser pointed up. So here's MHC. Okay, so here's our T cell receptor. Here's our MHC class 1, and you can see it's class 1, and here's our MHC class 2. Here's antigen. Antigens are red. And then, so they're presenting, they have MHC class 1 and class 1 and class 2, but they also have these little molecules over here that's the co-stimulatory molecules. And so they're receiving signal 1, they're receiving signal 2, and so now these are going on to become effector cells. These are going to start secreting cytokines, and these cytokines are going to help to promote proliferation. And the key cytokine here, again, is being shown as IL-2. There are many other cytokines that are going to be made, but IL-2 is really going to drive proliferation and the state of these cells. And these cells are going to become helper cells, and these helper cells are going to help cytotoxic T cells, and then they're going to differentiate to become either memory helper cells or effector helper cells. And you want both. You want memory cells because you want to be able to quickly respond to a second attack by a pathogen. So your memory response again is going to be much quicker because these memory cells are out there waiting and looking for antigen.
Excuse me, when you say co-stimulating, they have a little, what kind of molecule are these co-stimulating?
So the receptor would be the APC would make CD80 or CD86, really talk about, because there were some of the earlier ones that were discovered,
CD8,
for
CD8.
CD80 and 86, and then the
For both,
receptors.
I think.
Huh?
For both, naive CD8 and the
Yep.
CD4.
Yep.
They both bind
So,
like
yep. So they both have CD. So, for example, here you have. Let's see, where's CD80? So CD80 is expressed on your antigen-presenting cell, whereas CD28 is expressed. So both CD4s and CD8s express the CD28. That's the receptor for CD8. So again, important points to know for T cell activation are you need two signals. The first signal is going to be antigen via MHC. The second signal is going to be co-stimulation. And in that case, I would expect you to memorize that CD80 is the co-stimulatory molecule expressed on professional antigen-presenting cells. We don't normally have CD80 expressed on other cells. And then the receptor for CD8 would be CD28. CD28 is going to then bind. So CD28 is on the T cell, CD80 is on the antigen-presenting cell. So that's an important thing to know who's on who. And then you'll also notice that CD28 can actually bind to other things. We don't have it in this picture. We'll talk about that. It can actually bind to molecules that will actually suppress proliferation. So suppressive molecules that are upregulated later on. So let's first. So again, what we have here is we have when T cells see an antigen, they will then localize more and more T cell receptors to the lipid rafts, and they will form what's called the C-SMAC, and you get a P-SMAC. And so in the so you get the central we call it a C-SMAC, a central supramolecular activating complex. And we call it that because all of these T cell receptors all kind of get tightly organized into that central area. So in that, if you look at that central area, if you stain it for CD3, you're going to see lots of CD3, lots of T cell receptors in there. Whereas in the periphery, now you have mostly your co-stimulatory molecules. You have your also you have molecules that are adhesion molecules. So our LFA1 and ICAMs, CD2 and LFA1. So you start to get adhesion molecules. So they kind of hold it together. They kind of hold those cells together. You have adhesion molecules, and then in the center you have lots of interactions occurring. And if you get enough signal, so that's kind of helping to, it's like if I want to amplify the signal, say I'm looking for a radio signal and I hear it, then I want to amplify that signal. So in order to amplify the signal and see if they're really getting something, they want to get more and more T cell receptors and more MHC molecules all localizing to the same area and kind of congregating and saying, hey, how much of this antigen is there? How much of a signal am I going to receive? If I receive a strong enough signal, I'm going to, signal one is checked. Then they're looking for co-stimulation. If you receive that co-stimulatory signal, that's checked too. Now, if I only receive signal 1, but don't receive signal 2, then these cells will become anergic. And as we said with B cells, and it doesn't necessarily mean these cells die, but they're not going to proliferate, and they're going to kind of not respond to antigen. So they have to receive both signals. So they may become apoptotic, but they may just become anergic and then go off and not respond to antigen anymore. So in this way, if you have T cells that see antigen, but they don't see it on the antigen-presenting cells, let's say they come into contact with antigen that they recognize, that they bind to, but it's only on a cell that doesn't have co-stimulatory molecules. It's just on an epithelial cell. They're going to become anergic and not then respond, not mount an immune response. It's only if this antigen has been taken out. And this is kind of a way for us to regulate whether we really want to turn on a T cell. We don't want autoimmune disease. So there's always this balance between, do I want to attack it or not? The cells have no way of knowing self from non-self. Their only way they're knowing self from non-self is by having lots of checks and balances, keeping them in check if it is self. So checks that something is self it's presented a lot. It's in the periphery. It's on epithelial cells. I see it a lot. I don't see it a lot in antigen-presenting cells because these cells are dying. They're not sickly. They're healthy cells. They're not being phagocytosed. They're not becoming necrotic. They're not, releasing antigens off into their surroundings. And so therefore, I have no reason to respond to these cells. To a cell that has self-antigen doesn't have a pathogen in it. But then, and that's where things like chronic inflammation can lead to autoimmune disease, because now you start, and that's why they say you should take care of, if you have inflammation, or you have a wound that's not healing, or you have anything that is a recurring thing, you want to take care of that, because you don't want constant damage happening to the same tissue over and over again, because that increases your potential to have a T cell that's now going to respond to the self-antigen. Because if you continue to have tissue that isn't infected with the virus, that's not necessarily, that can actually lead to, damage to tissue can lead to release of self-antigen. That self-antigen can be taken up by antigen-presenting cells, and then it could induce, because it has signal 1 and signal 2, it could induce an immune response. So things like chronic inflammation, it could be virally induced, but it can induce autoimmune issues because your cells are constantly releasing self-antigen because of constant damage, constant necrosis.
Maybe that's why people who don't actually have celiac disease because of a sensitivity to gluten, see a progression of their sensitivity more to eat it. Because I know some people with celiac if they're sensitive to gluten, but they go a long time without eating it, and then they eat just a little bit, it's not as bad as if they had eaten it never.
That's what I think we're still trying to figure out, I guess. Allergies are still one of those complicated issues of how do we induce, because in general, we're supposed to induce tolerance to things we eat. We have our gut, we have our, the cells, the B cells and T cells that are in the gut, and those tend to, as long as there's not tissue damage, will drive tolerance. So something like that, it's interesting that those people are becoming intolerant to that antigen. So it is potential that maybe that antigen is being picked up, presented by hfcs. And that one I don't understand very well. I have to look that one up to see if we have any inkling as to what cells are involved. I don't know if that's a B cell or a T cell. A lot of allergies are B cell, but not all of them.
Do you have
So if you find that anything like that is interesting, if you find that it's, if you see anything in the literature that says it's a T cell induced response, that would be interesting to look at.
Dr. Thompson,
Yeah.
can you put the chapter 11 and 12 up?
11's up right now. I'll put up 12.
Yeah, because if you have a Monday, I had to work a whole day on Tuesday. I don't think I have any time to study for anything.
Well, go through chapter 11 and 12. I'll put up 12 when I get back to my office. I'm going to stop here because I'm actually over time. But just start thinking about T cell activation, how you need T signals. You need that co-stimulatory, and that's to help kind of prevent autoimmune disease. To kind of say that, hey, these professional antigen-presenting cells, these are the guys I want helping them mount immune response. I don't necessarily want any old cell in my body inducing a strong immune response, because I may get an autoreactive response. That's the case. So these cells and the professional antigen-presenting cells are kind of just constantly going around, picking up antigen, going to the areas of inflammation. They populate our skin, looking for pathogens that are coming in, and so they're pretty much everywhere. And just picking up antigens that they find and presenting them. And so factor to know is that T cells, if they don't get that right signal, they will